My name is Nonappa and I'm associate professor in chemistry, particularly in nanochemistry. I have a history of working on nanoparticle for more than 15 years. And today, based on your answers, even before your answers, I had certain assumptions that why are you are taking this course? And the first one, maybe you have not studied microscopy, and it's evident some of you don't have any knowledge. It's not important. But you are interested to know maybe what kind of opportunity the microscopy can offer for you. And the second is that you have access to maybe microscope, but you don't know how to use it. Maybe you have your own microscope and you don't know how to get a good image. Or you have a background in physics, optical physics, chemistry, or nanoparticles, and you are not really interested to know anything about this, any complex things, because it's too much. But still, you want to know something. Or you are simply interested how it works and how to use them. And as I already saw, at least one answer, just for fun, that is the best answer for this course. If any of these are true, then you are in a very good lecture today. And what are we going to do? At the end of this lecture, and very first thing is that you don't have to remember anything in this lecture. We will try to understand by doing. So there is no need to remember. Don't put your energy to remember, but let us understand how things work. So we will look at some of the historical development, some basic understanding of microscopy, the principles. Then I go a bit more advanced because it's a nanochemistry and nanoparticles. I will go a bit more into how electron microscopy works, and then we look briefly about atomic force microscopy. These are the most advanced techniques used to study nanoparticles. If there is time, I can show some selected example and a bonus part of how to use some even more advanced technique to study in multidimensional microscopy. So before we go, many of you know a human eye. That's how we can see, you know, the physics behind this or biology behind this, which is very complex. But the most important for us is the lens and retina and the object, where this object is placed. Now, depending on the size of the object, distance of the object, and the visualizing angle, you are able to see an object very clearly, or you are not able to see it. And now, any of you know what is the limitation for us to see? What is the size limit? What is the smallest object that we can see naked eye? Any answer is fine. Don't be shy. What is the smallest? The smallest object we can see is a human hair. And average diameter of human hair is 18 to 100 micrometer. This is a high resolution microscopy image. It's an electron microscope image of human hair. If you take the diameter of this is about 100 micrometer. It can vary depending on many factors. Now, if you want to see an object, as I said, we have to look at a very sufficient visualizing angle. The reason is that human eye, we cannot change the lens shape or the size, and that's not possible. And if you bring an object very close to our eye, you can try this in a letter. You bring it very close to your eye, you are not able to see anything. You will see blurred structure. Why? Because we have some limitations. So therefore, it is recommended, for example, when you read, it's a 1 feet distance, not very close, not very far. That is when you have a normal vision. But of course, there can be short and far-sightedness, which requires then correction. Now, we can see objects within the visible spectrum of the electromagnetic radiation. We are not able to see anything in the ultraviolet or infrared, but there are exceptions in some living organisms, bees and snakes and sea. They can see in UV and also in IR radiation. That is extraordinarily important. We cannot see some pattern, but bumblebees can see these patterns. What we see when we look at an image is two things. One is brightness, the other one is a contrast. So an image should be presented in a brightness and a contrast. And that's because we have some specialized cells in the eye, cones and rods. They have specific function and they are responsible for vision at specific wavelength. And when we present an object or a color or a figure, we also have to remember there is significant population who have some deficiency of vision. That is called red and green color or blue or yellow. This is less common, whereas red and green is more common, color blindness. Though it's not a blindness, it's a vision that is not normal. What is the simplest microscope? And then why we need microscope? Why we need microscope? To see small things, yes. So to see small things, things that we cannot see in naked eye. So that's why we need microscopes. So the name itself says that micro size, like the limitations beyond the eye. The simplest microscope is a magnifying glass. If you take a magnifying glass and if you have an object and if you look at it, you will create a virtual image. And that's this image we cannot capture in a film because it just looks as if it's on the same side of the lens. And we can visualize this, of course, but we cannot capture. Now, instead of one lens, if you have two lenses, this becomes a compound microscope. Now you can make a real image. This can be captured in cameras or photographic film in olden days. Now, if I invert this, it also becomes telescope. And telescope has developed very fast compared to microscope. So how this entire microscope has developed? It has a history of more than 600 years. And in around 13th century, spectacle makers in the Netherlands were making really highly precise glasses and spectacles and lenses. Then Galileo also used the telescope. And of course, he did say microscope word was used, but his main invention was telescope. The word microscope was really coined by not by a scientist, but an artist called Giovanni Faber. He was drawing some beautiful images, looking at these objects using lenses, plants and bugs. But really, some breakthrough came in the 1870s when Abbe proposed this diffraction limit. That means what is the limit to see an object or details in object or separate two objects? In 1931, the first electron microscope was developed. So then you can go beyond light microscope resolution. You can see even smaller object. There are several Nobel Prize. Nobel Prize was awarded for this 1986. And then there is more and more. There are two more Nobel Prize for cryo-TEM and also for scanning tunneling microscope and AFM. We will look at all of this. Let us look how it has developed. So Robert Hooke had the compound microscope. It's very simple. You have the lens. There is an objective lens and eyepiece. And the light, there is a lamp, there is oil, and a water containing a water container that will focus the light to this lens, and then you have the object here and you can visualize this. He used these cork cells, he called cells. These are not cells, these are not just compartments. But there was a problem because the objects, if you look at the images, good images are found only near the center of the lens. As you move away, then you see what's called aberration, and you are not able to focus and get good image. Then Anton Leeuwenhoek, he made a single lens microscope. That means a lens which is 1 millimeter already in the 18th century. You take this lens very close to your eye, you can magnify an object up to 400 times. That's really a big improvement. However, this is not very convenient because you always have to take it very close to the eye and see an object. He was able to draw bacteria and many other small objects. Again, the compound microscope came Robert Brown. He used the onion cells and was able to visualize nucleus. This is the history. So what are the tasks of microscopes? Anyone want to tell me? 3 tasks, yes. Magnifying, yes. Anything else? Resolution, then? What is a resolution?
For example, there are 2 objects, so you see like the border.
Brilliant. Yeah. How to differentiate or distinguish 2 objects. So yeah, produce a magnified image. That is the first task. Then you want to separate the details in an image, that is the resolution, and render the details visible to the human eye or camera. So these are the 3 simple tasks. You want to magnify, you want to see, and you want to capture them. This is what every microscope does. Whether it's a simple compound microscope or the more advanced electron microscope, they all have 3 tasks. You want to visualize, you want to separate the details, and you want to capture them. And then you analyze. Only difference is they all have lenses, and the lens may be quite different. They have a source of light. The source of light can be different. The way the images are captured can be different. But the overall principle of all microscopes is same. Then in 1880s, because there's some light microscope, the resolution of microscope, I will come to that. Abbe came up with a formula. According to him, the resolution will depend on the wavelength of light. Do not remember this equation. All we want to know is the wavelength of light. And that means if we have certain region of electromagnetic spectra, we can calculate what is the theoretical limit, that how small object can be seen, or what is the smallest distance that can be resolved between 2 objects. That is the resolution. I have 2 sticks, but I want to know how far they are. So the best resolution, if you put a 400 nanometer here, now you will get 200 nanometer is the resolution for this wavelength. Now, this was another improvement in 1920s, where the duality of wave-particle theory has appeared. Louis de Broglie in 1924 proposed that electrons can behave both like particles and waves. What does it mean? It means it is also much like a light. Now the idea comes, can we use a beam of electron instead of light? Because electrons can have smaller wavelength. Smaller wavelength means higher resolution. But the challenge is how to use that, right? So electrons have certain wavelength. You can use all this equation, but now how to use this electron and create this beam? And electrons, as you know, in the lamps, if you increase the voltage, you see brighter. So if you use higher accelerating voltage, can we make even a huge beam of electrons? So then the wavelength can be decreased, higher energy, lower wavelength. So you can use, then you know what is the wavelength. Then you can calculate this always in kilo electron volt. You can go to picometer. That means you now go to the resolution into atomic scales, nanometer atoms. You start to go from a naked eye to 400 million times more resolution than a naked eye can see, and 4,000 times more than a simple optical microscopes can see. So this is the whole idea. There's a basic concept. There comes the quantum physics and mechanics, wave particle theory, and then use the same thing, and then you start to see. But there are challenges still, right? Because electrons scatter in the air. That means you have to prevent that, and it's not so easy. However, in 1931, the first electron microscope was built in Berlin by Ernst Ruska and Max Knoll, and after 55 years, they got the Nobel Prize. And electron microscopy has revolutionized almost all the field today. And first, they used magnetic lenses. We will come to that. You will see what is a magnetic lens. They were able to see the same resolution as a good theoretical limit that you see in optical microscope. They use a third lens, they reduced by half, and later on there also comes scanning electron microscope. Now we have seen the basics, the history. Any questions here until now? No. All clear? You have understood? Great. So now we go. Now the fun part starts. You are going to use your laptop or mobile. I recommend you laptop. What I have given here is a virtual scanning electron microscope. I will also do that, and in case you have a problem, you can use this URL if you are using laptop. You can also scan and get the URL. This is the same. All this lead to the same web page. It's a virtual simulator, and I will also go through that. Yes. Now you can see here, this is a virtual scanning electron microscope. And what you see, here comes the instructions as we go on. The first, it says that let us select a sample. So there are many things that you can think, but we can select maybe some butterfly wing because it's very beautiful. So let us take this. Later on, you can practice. What you see in this here, the right side is kind of a schematics of what actually happens in the microscope when you do. And now it says load the sample. You can watch here what happens when you say the load the sample. Yes, the sample is loaded. This is a sample stub where there is your sample is here, thin segment. Now there is an instruction. Okay, evacuate. The idea is that you want to have this chamber without any air. It is under vacuum. So let us evacuate. All are fine? It's going fine? Okay. Okay. Don't hesitate to ask me. So let us take some electron volt. It's a kilovolt I was mentioning. So it's a 5 kilo electron volt. Spot size, it's about the size of the beam. So we can take, say, 5 nanometer if you want. Let us select the Z height, is the height with the sample. And then you need high tension. So switch on this. So you start to see something happened here. We will go through this step by step. A beam of electron passing through this, and then now it is actually deflecting. So we have an electron source, we have an anode plate, we have condenser lenses. These are magnetic lenses, and then there is a deflector that deflects this electron beam very sharp and narrow. And what we have here is a detector. So there is a butterfly wing. We can see some blurred image behind, but now I said it has to be presented with certain brightness and contrast. So we have to change the brightness and the contrast. Now by changing this, you can visualize, and then you also have to focus. Are you able to see? Yes? Okay. And then you can increase the magnification. You can increase higher magnification, then focus. And you can go higher. Higher magnification structures can be seen. There are details coming. It's a real image. And if you change the focus, you see it can blur. So you can increase the brightness if you want. And we go higher magnification. You start to see it's a real insect wind. This is not simulated. This is the data acquired in Microscopy Australia. Many universities together and Thermo Fisher Scientific developed these virtual tools. Now, if you really want to play with this, you can change the electron volt and you can change many aspects of this. But the whole idea is that now you know what to do and how it works. components of this microscope. So these are the few things that what actually are important in scanning electron microscope. The first one is electron, then filament, electron cloud, anode plate, magnetic lens, sample, deflector, detector, image formation. You have done this already now. So, but now we can go and look at electrons. They're always there and they're moving around the nucleus. Now, many of you know that if you have a lamp or a bulb, there is certain filament, right? There is a tungsten filament, for example, and depending on the voltage, you can increase or decrease the brightness. So if you know this concept, you already understood what is going on in electron microscopy. And instead of that kind of lamp in electron microscope, we have this kind of a bent wire, which is called filament. Now, if you increase the voltage, then in this region, we start to get some thick cloud of electrons, because as you increase the electric current and increase the accelerating voltage, electrons, you push them away. Now they can pop out of this wire. Now, if there is a very high accelerating voltage, you start to make a cloud of electrons. And that's why there is electron cloud. So there is electron, the property of electron, how they react when you have electric current is used, and use a filament and electron cloud. Now electron cloud can escape. So you have to prevent this escaping from escaping. So what you need? You need something opposite. Electrons are negatively charged, right? So you need a positively charged something that is called anode plate. And this cloud can be then pulled and directed. This is exactly what happens when you switch on the higher voltage. And this then directed towards your sample. And now what we need, still it is not enough. You have to condense this beam and focus to the sample. So you have this magnetic lens. So these are nothing but coiled wires. Now we pass the electricity, then it generates induced magnetic field. So that's why we cannot use glass because of the high energy electron radiation. So you condense, you have lenses. Now it's not enough. You need to scan your sample. So that's why we have a deflector. Again, this is an anode deflector that allows you to scan the sample. That's where the name scanning comes from. There is scanning, there is electron, there is microscope. So there is scanning using a beam of electron. You have your sample here. Now, when it interacts with the sample, there are many phenomena can happen. The first is either a sample just burns because it's high energy, a really high energy, or your sample may produce some secondary electron. It may get charged, or it can get some backscattered electron. These secondary electrons are the most important one, where then can be used. Now, how you get this image? You see some dark, some bright, some gray in the SEM image, that in the insect wing. There's a black background, you saw some gray area, then you saw bright area. When there is a black, that means there is no interaction or there is no secondary electrons are produced. So here, these secondary electrons or backscattered electrons comes, and you need a detector to capture it. So now we need a detector that can do capture and give information for the image formation. Told about the deflector, this is the anode deflector. The sample is there, it is scanning the area. As it scans, it identifies the areas where the sample is there or some structures are there. Now, when there is a sample or some specific structure, there's a particular number of electrons are generated, which are captured in this detector. So it detects, and when there is a black, it means there is no sample or no structure or no detail. When there is a gray, that means there are some, gray means maybe there are little, bright means there are a lot of structural details. So it distinguishes, because when the sample interact, then it has to detect where the structure or details are there. So this is what you have seen. There is electron source, gun, we call also as a gun. It can be filament, it can be many others these days. Anode plate, lenses, multiple lenses, a deflector, sample is here, and then the secondary electron image. Any doubt? Deflector is here. It is also a kind of anode deflector because the beam then is sharp because it's anode which is differently charged. So it has a different role than this anode plate. Deflector is needed because otherwise the beam, you cannot scan the sample. So if you are using microscope, then there is something that you have to make sure that its deflector is working correctly. Yeah. This is very simplified, so you don't have to remember. But because you have done, you now know like all the things that happens. You go back and then you practice. So we can also now define, because this is a IUPAC definition, but I simplified this. What we have seen now. So how do you define a scanning electron microscope? Very simple, because you have already seen now what it is. It uses a beam of electrons to generate secondary electrons from the sample. And to lesser extent, it also creates backscattered electrons. Then there are different detectors. It uses a very finely focused electron beam, which is about few nanometers or tens of nanometers. And what we get is a high resolution image and high depth of field image. It can mislead like a 3D image, but not really, because it's still surface, but the depth of field is high. Any questions? Is it clear? SEM? Yes. Okay, I go on. You can stop me if it is not clear. Now we can compare the simple light microscope and scanning electron microscope, very simplified. Scanning electron microscope can come with very advanced, with a price range ranging from $500,000 to up to several millions. So that's because of the complexity. It has to work in a high vacuum, and the entire vacuum system is called column and sample chamber. So same thing, we have lenses, we have eyepiece, objective, but we have also lenses, but these are magnetic lenses, whereas instead of simple light or LEDs, what we have is a electron gun or beam of electron used. So as I said, the complexity increases, and then at the same stage, the resolution that you can see also higher. Fine, no questions. So how then this image is done? So if you want to image, say you have a sample, so there are a few simple things we need. You saw already this sample stubs. You need some adhesive tape, because most of the organic sample if you use, they're not good conductor, and you need some heavy elements. So you always have to coat. So use some coating. It can be carbon or conductive coating, gold, palladium, and these are stubs look like. These are maybe few millimeters or centimeter almost, I think it is. And then you can directly use any types of stubs. Any types of material can be imaged. There is no limitation except that you cannot use water. So if you for example, here is a gel. I show only a couple of examples because you can see already examples from the virtual simulators. And this gel, if you look, of course, is macroscopically very transparent. But if you take a scanning electron microscope image of this dried sample, freeze-dried, you will see about few tens of nanometer fibers highly entangled, where these pores represent where the water is trapped. So you can see high resolution image nanofibers. If you take small molecules, so I think there is also self-assembly in this course. These are small molecules which can assemble in different solvent. But if you look at the scanning electron microscope, you start to see these fine details, screw-like structures or helices or twisted tapes. So these are the things. So the things that you cannot see in naked eye, which are under scale of nano to micron scale, that can be visualized by electron microscope. This is not the only thing. You can also detect X-rays from this. You can do elemental mapping. there are more advanced techniques. Why don't we take a challenge? Yeah. Okay. Now you have seen scanning. The next we go transmission. As the name suggests, there is also electron. It's also electron microscopy. It's an electron microscopy technique. But then the mode that we see is not scanning, but it's a transmission. It is also possible in this technique to use scanning. Then it becomes scanning, transmission, electron microscope. We don't go into that. But maybe we already start with a virtual simulator because it is easier in the same way. And for me to explain, it's much easier if we start with this simulator. I will also show you. It is bit challenging, or not really challenging, but bit challenging compared to scanning electron microscope. So, but exciting. Yeah. So what you see here is the structure or schematics of transmission electron microscope. Very complex, but simplified version. And what you see here is the same structure. We have a beam of electron. And then there are a lot of lenses, more lenses than what that is in a scanning electron microscope. We have a sample holder. And then there are detectors, there's camera, digital CCD, or direct electron detector. So first thing, there is instruction you see at the top. You have to now first evacuate and remove this holder. So you saw the holder is here. There is a tip of this holder where you insert the sample. You can choose any of this sample, but since this course is on nanoparticles, let us choose nanoparticles. And now everywhere you have to click the button. It is highlighted with yellow color or yellow border. So the sample is already here in this. Now we have to insert. And once you insert, it's the same because it will evacuate. And we insert the sample further. And you see some changes in this place. So this is good. You can, if you have possibility to keep this aside, you can also close, but you can always get it back by clicking diagram. Now we will apply certain accelerating voltage. It is 200 kilo electron volt here. And then there is just focus now here. Let's switch on the beam. The beam is on. When the beam is on, you start to see also the beam here. This is a transmission electron microscopy grid, TEM grid. It is made up of copper or gold or nickel. It can be also silicon nitride. And these grids have very thin membrane where your nanoparticles are loaded. And these membranes are of up to 30 nanometer, 5 nanometer, 30 nanometer. Now here there is an highlight which is called beam shift. That means you have to bring this beam very close to. Are you following? Yes. Great. So now you can bring it very close. And it has to be center. When it's center, this highlighted border will disappear. Now it says brightness. That means you have to spread this beam around this entire black area. Yes, now it is fine. Then you go and select some. At this stage, please also look at this. The beam is now on and beam is going through, and it's kind of. Then you have to put some aperture. Oh, yes. Okay, I have to restart this for some reason. Let me do. Yes. So once you select the aperture, you have to have this diagram here. So you have to press insert. You see this, the color which was with blue has now changed, and this condenser aperture is now inserted. The next is to choose the aperture size. We can choose small, medium, large. Let us select small. And now you see there is something, the beam is not circular anymore. It has deformed, and this is called stigmatism. So we have to make it astigmatic. First, you move it to the center. Yes, and now you make, so if you click in the wrong direction, it becomes worst. You have to make it circular by clicking this. Yes, now it is circular. Now increase the brightness, spread the beam. Choose a magnification which is low, and we start to see. And we have to also make sure that sample is not moving too much. That's what we call a wobbler. And you have to minimize this movement. So now the sample is stable. Click back, you say. One thing is routinely used is also looking at the diffraction, which is, we will go through that bit later. Then you insert again and routinely take this sample beam back to the center. Now you are all set for imaging. You just click imaging, and it's a part that you are interested. These are nanoparticles. These are real image taken. Now in this you see a lot of these nanoparticles, but you want to maybe look at those area. Then you bring that area to the center. Any problem? Are you able to see? No? Don't hesitate. If there is a problem, tell me. Yes. Now you have small, we can go a bit higher magnification. And again, bring it to the center. Bring it to the center. Okay. Now we can go high magnification, even high of a single individual nanoparticle. And that blue circle will come to the center. And now you do some focusing. Find the correct focus. Okay, maybe this is. Now go to the diagram here. And you want to insert camera. So we are in this place. Camera is inserted as you saw. Now we can take the image. If you want, you can acquire this image. And let us download it somewhere. Yes, so here is image where you can see individual atoms. And you can change if you want to a different materials. Then the instruction says if you want to continue to the next step, we are not in later. But the idea is that you have seen how this is done. So this is a bit challenging. So if you look at the anatomy of transmission electron microscope, we have electron gun, same, there is an anode plate. Then we have a lot of lenses, huge number of lenses. But unlike in SEM, the sample is located here. And then it is transmitting, so that's why it's a transmission. So you get a transmitted image or projections from this. Many things happen when electron beam interact with your sample. It can emit X-rays, so you can do elemental mapping, or it can generate backscattered electron. That is the principle of electron scanning electron. It generates elastically scattered electrons, and this is the what we see in the diffraction. And these are the transmitted electron. That's where the image is coming from. There is also inelastically scattered. That means it depends on the elements. So then you can use energy expressive loss spectroscopy. So in transmission electron microscopy, we are not just looking at material morphology or structure. We can look at their X-rays like elemental mapping. We can look at the surface topography. We can look at how the structures are by diffraction pattern, just like X-rays. Or you can do the spectroscopy. That means almost everything that can be done instead of multiple instruments in a single instrument, and you need extremely small amount of sample. So that is the beauty of this. It's not just imaging. So we are moving more towards imaging to real structural characterization. What happened in this process when you look at this simulator? So what is then transmission electron microscopy? It's an analytical technique for an electron transparent sample. What does it mean, electron transparent? Anyone? What is the meaning of electron transparent sample? What happens in a transparent sample? Yeah, it goes through. Yeah, that means the sample where the electron beam should go through. So that's one of the limitations. Sample has to be thin enough for this. And then what happens? When the electrons are transmitted, there can be scattering, multiple or different types of scattering. And this is one of these phenomena is then recorded. Because the sample electron beam is going through the interior of the sample, so we get the internal structures. But it is only two-dimensional. It provides a very high resolution image. We can see atoms. And I think I have some images of high resolution I will show you. So here is a light microscope. Here is a scanning electron microscope and then transmission electron microscope. We see the complexity in the anatomy of microscopes are going. At the same time, we also see the resolution is increasing. From nanometers, we are looking at atoms or even at picometer level these days with a very expensive. So this high-end microscope can go from 2 million to 10 million euros. They are so big, they cover an entire building and highly sophisticated. But how these samples are made? And you have seen when you say loaded the sample, there was a grid-like structure in the holder. So there is a small grid, it's a 3 millimeter in diameter. There's extremely thin membrane. It can be a polymer, it can be a carbon, graphene in these days, or silicon nitride. then you need less than a microliter or nanoliter of sample, but we generally use 3 microliter, blot out excess, and you have a very thin sample loaded. And we can get very high resolution. The gold nanorods, and you are seeing here individual atoms of a gold nanorods. We get very fine details, internal structure, morphology, atomic level details, and more, even more.
The fine killers as well.
This is just to look at the possible screen and to move the samples. These days nobody uses this binocular. This is still, it is there, but it's very good. Like if you're a good in microscope, I like to see in binocular because it really helps a lot to move the sample and find the correct thing. But there's a phosphorescent screen. It's not used these days much because your instrument and operator rooms are quite different these days. It's more sophisticated. This has multiple detectors. See, there is a STEM detector shown in this side, but there are like multiple detectors. In our microscope here, unfortunately, the reason why I am showing this virtual simulated today because of the construction, we cannot use our microscopes on weekdays. And weekdays maybe after 8 p.m. or only on weekends. So it's impossible for you to have a hand-on experience or look at them. Looking is possible, but do anything or see how it works. But what you see is the same way how it works. We have detector for imaging, and we have electron detectors, scanning transmission mode detectors. We have detector for EDS like elemental mapping. We have Heels detector, and there can be 4 to 5 detectors, different detectors. They detect different types of information coming from the sample when it interact with the electron beam. Yes. Any other question? So what else can we do? We can study many things. See, here is one example I'm showing. It's a very small nanoparticle of 2 nanometer. And then you can self-assemble because self-assembly is one of the topics here. And this forms this kind of hexagonally packed structure. And these are nanoparticles. They are very well defined, these kind of structures. They self-assemble. You can see the hexagonally packed structure. You can determine the exact distance between these particles, thickness of this. So then what else? Can we visualize particles? Here I am showing one of the studies done also here in our PhD student Abbe recently. These are the 2 nanoparticles. We irradiate with the electron beam, and they are, let's say, a few nanometer in distance between them. And this image is taken for 1 hour continuously. I have fast forwarded it by almost 60 times. So the particles, when they interact with the electron beam, they can also undergo some damage because we are talking about electron, which is negatively charged, high energy. It can eject some electron, create surface charge, or burn them. But they can also make some new structures. Here the particles are coming together. As you can see, they come together and create a new structure of merged structure, which we call coalescence, or they coalesce. Or in another video, I can also show how the particles can repel. That means they run away from each other, they go away from each other. So these details cannot be imaged in any other microscopy technique. And we can image directly and see even at atomic level how the particles are coming together, forming a new type of structure. And here it is shown that the particles first come closely, but then they start to go away from each other because of the charge generation. It's a highly sophisticated method where we can today look at how atoms are behaving. So that is the beauty of high-resolution electron microscopy. So, but there are limitations for this. The limitation is that if the sample thickness is much more than, say, 50 to 100 nanometer, then it's very difficult. Beam of electron will not go through. It is no more electron transparent. We don't get good details. Then it works at extremely high vacuum, all electron microscope, because electrons scatter in the air. We want to prevent the scattering only by creating high vacuum. So your material that you study has to be beam tolerant. If you take electron microscopy course, one and the most advanced electron microscope, there is a dedicated part also about beam damage and many things. I'm also teaching a couple of lectures there. Any questions for TM? I will have some more details if needed later at the end of this. I think we quickly go through AFM. This is another topic. Again, there is atomic force microscopy. Something has to involve with the force. Again, imaging. So it's also rather interesting. It's another Nobel Prize winning technique of 1986, developed in 1981 in IBM. So IBM Labs in 1981 first developed the scanning tunneling microscopy using the tunneling effect. But then it led to a series of or family of microscopes called scanning probe microscope. Atomic force microscopy is one of those microscopes, and what it is able to give very high resolution surface topography of a picometer scale, quite different. And how it works? It has few important components. The first one is a cantilever. And this cantilever is about 200 micrometer long, and it has a spring constant because it has to bend of about 50 Newton per meter. Then it has a tip. This is often you hear AFM tip made up of silicon nitride or silicon, which the tip radius can be 10 nanometer, 20 nanometer. This shape can be changed, modified, surface coated if needed. It has an apex diameter of 10 nanometer, but the height from the length or height of this is about 4 to 5 micrometer. There is then a piezoelectric scanner, there is a laser beam, and then there is a photodiode. So when the sample, the principle of it works because when this tip, a sharp tip, goes very close to the object, and on the surface of your sample, there is a overlap of atomic orbitals, so that leads to repulsion. When two materials come very close together, then that leads to repulsion. That's where the force is. And then this repulsion can be measured because a beam of electron falling on the cantilever that changes in the height as you move on will be detected using the photodiode. Then the image can be processed using computer. Principle is simple. Again, the force is involved when the tip is interacting with the surface atom. Because of the atomic orbital overlapping, there's a repulsion created between these two objects. One more, and this will be the final exercise for you to take it to AFM. Yes. This is simple. So here you see the same structure. So there are different modes how this AFM works, atomic force microscopy, contact or tapping. Let us now take a tapping mode. Are you able to see this? Okay, great. And then again the same, that you see this laser beam, right? And here is the laser beam, here is a cantilever, here is a photodiode, and your sample, and there is a piezoelectric scanner sample stage. Now your goal is to move this laser beam to this exact position where it's supposed to be. So let us move it using this, or maybe, yes, and very close. Ah, yes, almost. Yes, now it is there, but it is not in the correct position. So now we have to use this, move the photodiode a bit. And you can see this in the right side already. This is exactly how it is in AFM. If you go to AFM and see it. Yes. Okay, now the instruction might say that there is in the target amplitude, it has to be 500 millivolt. Yes, it is done. Then you have to select certain frequency in the range. And then auto tune. Scan size, you can choose the area that you want to scan. Say, let us take 10 micrometer. And there are some details like what actually happens if you choose high or low sample scan size. Then you can choose something. Scan rate, that means how fast you want to scan. We can choose, say, 5 Hz. It also gives some caution if you are using some values that might damage. Problem?
Yeah, if you go too fast, you broke the demo.
Yes, so don't destroy your instrument. Yes. Yes. It requires some gentle way of operating. Yeah, then you can use some set point. It doesn't matter what is, because right now now you have to move the motor. This is where you crash. And you have to be very careful. You don't go very close. And yeah. Yeah, now you see this engage, because generally you go close, but because our human high resolution is not so great, right? So we want to do the instrument some work by itself. And here it is. And now we start scanning. And you start to see your AFM image. And you can change now if you want the scan size, the frequency, the area that you want to scan, and accordingly you can get high resolution structure. So you see some objects in this microsite. We can disengage if you want. We can go to contact mode, and how the contact mode works. Let us see if we can. Then you see also the difference between two modes. Yes. Scan size can be 10 micrometers, same as before. We can change it. Maybe 2 Hz. Keep it low. And let us bring the stage very close. Yeah, and then engage and scan. You see the difference between now the tapping mode and the scanning, the contact mode. We can take smaller size, then you will see the details. Or if you want to take 20 micron. Related to the tip diameter, we can also measure spectroscopy there, which is called force spectroscopy. We will not go into details. Here are some examples how beautiful structures you can see from AFM. These are protein self-assembled structures or peptides called amyloids, which has relevance in some neurodegenerative disease. And what you see here is the helical pattern of few nanometers of these fibers, self-assembled fibers, and how nicely you can see the helical pattern, and you can calculate with very precisely the pitch length, the distance between two helices. And yeah, and here are synthetic polymers, the polymer which have a lot of brushes. When you assemble them, they form these worm-like structures. You can see also the polymer brushes here, the worm-like structures. Maybe it's not possible in many other than TEM and AFM. And AFM, you get the beautiful topography. So you can really see the structures, any structure that's made or naturally existing with the AFM. But what else we can say? I said you can see picometer. Can we map a molecule? And this is an example. So mapping hydrogen bonding with the weakest inter and intramolecular bonding. So supramolecular interactions. These are this four-membered ring where which is a thin film made, which is a monolayer, that one molecule thick, and then you map and you see the intermolecular interaction. That's the interaction between two different molecules through this oxygen and hydrogen, which is attached to the nitrogen. So this is the real molecular structures mapped, mapping a molecule, and that means mapping an atom. So that is the beauty of